Ahora tendré el gusto de presentar al doctor Mark Brown. El doctor Mark Brown es un experto en investigación relacionada a nutrición de la Cleveland Clinic, es coordinador de uno de los principales laboratorios en el Lerner Institute, que es la parte de investigación de la Cleveland Clinic. Con ellos estamos en el proceso de desarrollar un esquema de colaboración, de intercambio de metodologías y de personal. Es su primera visita en México, por lo que agradecemos en especial a su presencia en este evento. Mark, welcome and thank you very much for coming to Mexico.
Okay, so thank you very much, Carlos, for the invitation and for everyone's attention to talk to you about potential collaborative research between the Cleveland Clinic and folks here. So I've had a wonderful visit. Carlos and team visited the Cleveland Clinic a couple weeks ago, and I think there's a lot of opportunity for collaborative research in the area I'm going to speak about, but many others as well. So today I'm going to talk to you about our research that's focusing on the microbiome specifically. So just to give you an outline of my talk, I'm going to introduce what the Cleveland Clinic is, just briefly, so you know about what type of research, both clinical and basic, is going on. I'm going to share with you a partly published and mostly unpublished story about coming out of this new center called the Center for Microbiome and Human Health, which hopefully we can find collaborative areas of research within the center. I'm going to talk to you about this microbe-derived metabolite, trimethylamine, and trimethylamine N-oxide, and obesity type 2 diabetes. And at the end, I'm going to talk to you about our efforts in drugging this pathway, so inhibiting the production of these microbial metabolites. So just briefly, a little bit of background about the Cleveland Clinic. It's a very large institution. Currently, we have over 50,000 caregivers. We see about 7.6 million outpatient visits, over 200,000 hospital admissions, and nearly 4,000 physicians and scientists, and it's on both basic and clinical sides. Also, we have a very strong training program with both residents and fellows. Although we're number 2 right now, we hope to move ahead of Mayo Clinic soon, so this is just the ranking from the U.S. World Report. The Cleveland Clinic is primarily known for cardiovascular disease, so we've been the leader in that in the United States for many, many years. So in particular with vascular surgery, actually 23 consecutive years we've been number 1 in the nation. We're also number 1 in urology. There are 13 specialties in the top 10, so we pride ourselves on excellent patient care. I myself am not a clinician. I'm a mouse doctor, so I can't speak to the successes there, but I'm going to tell you about some of the research. We're also top ranked in Ohio. So I'm not going to go through this whole list, but this is just some of the innovations that we've done both locally at the Cleveland Clinic and in some cases collaboratively with other institutions. Many of these are surgical, in particular the first coronary angiography. What I'm going to talk to you about today is discovery of this gut microbial metabolite and how it links to cardiovascular disease. And my lab is actually housed in something called the Lerner Research Institute. So like the other clinical institutes, this is the research hub. So there's actually over 2,000 scientists, including about 180 principal investigators. So these are people focused on model organisms, Drosophila, zebrafish, mouse models, rats models, all the way up to clinical investigation. So it really is a translational research place. And I just wanted to mention this because I know there's a big biorepository effort here as well. We have a growing Cleveland Clinic sponsored biorepository, a centralized biorepository, less so for diabetes, obesity, but more so for cardiovascular disease. But I think there's a lot of synergy with these biobankings. What I'm going to talk to you about today is the Center for Microbiome and Human Health. So I'm the director of research for this. So this is within the last 2 years, we've built a number of core facilities, which are already active and running, and hopefully folks here can make use of these through collaborative studies. The first is a mass spectrometry metabolomics core, so being able to measure, like you heard in the metabolomics in the last talk, being able to measure not only host metabolites, but small molecule metabolites that come only from gut microbes. We have a notobiotics core where we can house mice in germ-free, reconstitute them with different bacterial communities. Of course, the sequencing analytics core for 16S metagenomic sequencing, and this is quite unique where we have a culturing and engineering core where we can culture human commensal bacteria or a polymicrobial human feces consortium, but we can selectively knock out one gene at a time or overexpress it to do functional studies. So keep all that in mind. So here's one story of how we're leveraging this Center for Microbiome and Human Health to potentially treat human disease. So my training's actually in nutrition. I'm a PhD. So I have to tell you a little bit of background on how we followed abnormal lipoprotein metabolism actually to the microbiome. So my lab was actually studying a process called reverse cholesterol transport. In the context of atherosclerosis, you have these macrophage foam cells where you can efflux some of this cholesterol into these good HDL particles, which bring the cholesterol back to the liver. And then the cholesterol is ultimately excreted into bile and then into the feces. So this is what you would learn in a med school textbook about reverse cholesterol transport. However, when I was a postdoc, we found that actually if you divert bile away from the intestine, either surgically or genetically, the mice still excrete the same amount of cholesterol in the feces. So there's actually an intestinal pathway. It's called transintestinal cholesterol excretion. So I'm not going to bore you with all the details, but the same thing happens. HDL brings cholesterol back to the liver. A large proportion of that is secreted into bile for fecal disposal, but this TICE pathway is initiated here in the liver where the cholesterol is brought back into the hepatocyte, packaged onto actually an ApoB-containing lipoprotein, moves through the blood compartment, and then is taken back up through the intestine for excretion. So we think both of these pathways are active. However, there's multiple question marks about this intestinal pathway. We didn't know really what are the regulatory factors. So we had 2 mouse models where this intestinal pathway was either chronically or acutely stimulated. And in those days, we just did a microarray analysis. And what we found in the liver is what led us to the microbiome. So the most downregulated gene in both of those models was something called flavin monooxygenase 3, or FMO3. And FMO3 is a xenobiotic metabolizing enzyme. It's also mutated in a disease called Fish-Schroeder syndrome, which we can discuss at the end. But it was suppressed in both of these mouse models. And this was before I was at the Cleveland Clinic. So we made that discovery in probably 2009. And then this really seminal paper from Stan Hazen's group, who's actually my close collaborator now at the Cleveland Clinic, in 2011, Stan described that this pathway was associated with cardiovascular disease. So it's important to note the way he did this was through case control studies, cardiovascular disease patients, and untargeted metabolomics. So he was looking for what metabolites associate with disease risk and mortality outcomes. So what Stan found was this microbial metabolite called trimethylamine N-oxide is associated with worse cardiovascular disease risk. So this is a diet-driven pathway. Nutrients in high-fat diets, such as phosphatidylcholine or carnitine, are exclusively metabolized by the gut microbes. Enzymes in the gut microbes convert those compounds into something called trimethylamine. This goes to the portal circulation, and then our favorite enzyme, FMO3, converts TMA to this pro-atherogenic bad guy. So initially, my lab was interested in FMO3 based on the discovery through the cholesterol model. So we worked with a company, it used to be called Lyases Pharmaceuticals. That's not a very good drug company name. So they renamed themselves Ionis Pharmaceuticals, but this is a antisense drug discovery company, which we work a lot with for liver diseases. These are liver-targeted knockdown approaches. So we knock down with antisense FMO3. This just shows how well it works on 2 different levels of dietary cholesterol. This is the protein level, so it's essentially a knockout. And as a result, you accumulate the substrate of the enzyme, trimethylamine, in both dietary conditions. You get less TMAO, so That was expected. What we didn't expect, though, was a complete reorganization of host cholesterol metabolism. So when we knock down FMO3 on a low or high cholesterol diet, you can see massive reductions in stored cholesterol ester in the liver, no real effect on free cholesterol. And I'll just summarize because this is published, but this FMO3 enzyme is somehow intimately linked to host cholesterol metabolism. We think when we inhibit FMO3 that you're stimulating this intestinal TICE pathway. There's also robust reductions in intestinal cholesterol absorption, so bringing cholesterol across the enterocyte. And then there's depletion of cholesterol and oxysterols in the liver, which impact the biliary secretion of a number of different lipids. Okay, so I think most of the audience will be more interested in this partially published and mostly unpublished study, is that this pathway also associates with obesity diabetes phenotypes. So this was actually two cohorts at the Cleveland Clinic, either in preventative cardiology or hepatology. And here we're just looking at plasma levels of TMAO and the odds ratio of type 2 diabetes. So very similar to cardiovascular disease, the more TMAO you have, the higher risk you have of developing type 2 diabetes. And then in the initial studies where we knocked down the TMAO producing enzyme FMO3, we actually saw that there was almost no white adipose tissue in those mice, although we were looking at more cholesterol phenotypes there. So there was another group independently at UCLA, Jake Lucis and colleagues, have what's called the hybrid mouse diversity panel. So this is a bunch of strains, over 100 strains of mice, which have a different capacity to be susceptible to diet-induced metabolic disease. So this black mice is very susceptible when you feed a high-fat diet. This white mouse has no metabolic phenotype. So he looked at just metabolic, in this case, adiposity traits. It also tracks with insulin sensitivity. The higher level of TMAO, there's a positive association with adiposity across all those different strains. And then there was a third group, this all converging at the same time. We didn't know each group was working on this. This is Sudha Bittinger. She's in the Harvard system at Boston Children's. And what Sudha was working on was something called the liver insulin receptor knockout mouse, the LRCA mouse. So these mice selectively have the insulin receptor knocked out in hepatocytes. Therefore, they have selective insulin resistance in hepatocytes. And like Stan Hazen, she used an untargeted metabolomics approach to say what metabolites are different. And she found TMAO was the third most upregulated metabolite, and she found that our favorite enzyme, FMO3, was upregulated some 1,000-fold in these LRCA mice. And she also showed, using the same knockdown approach with ASOs, that this protected against some of the cardiometabolic phenotypes in those mice. Okay, so we followed up on this in a more traditional high-fat induced obesity model. So we knocked down FMO3 either in chow-fed or FMO3 knockdown mice. And here are the high-fat controls. They become obese over time. When we knock down FMO3, you're protecting the mice against body weight gain. There's not much of a phenotype in chow-fed mice. But if you look at adipose tissue weight, it's reduced in both cases when you knock down FMO3. And this is just an MRI image to show the high-fat fed control has a lot of fat accumulation. The mice eating the same amount but have FMO3 knocked down have much less fat accumulation. And when we quantify that, here's the subcutaneous adipose mass by MRI. There's a 50% reduction, and we were very surprised, but there's actually a significant increase in lean mass. So simply by inhibiting this microbial converting enzyme, which has several other substrates, we're changing the metabolic state of the mouse. And one of the phenotypes that we found when we knocked down FMO3 was activation of brown adipose tissue and beiging of white adipose tissue. I don't have the time to show you all the data, but in this case, we just took our ASO-treated mice in high-fat condition, and we put them in metabolic cages to measure oxygen consumption and heat production at 3 different temperatures, so thermoneutrality, room temperature, or cold, to stimulate thermogenic responses. So regardless of the temperature, you can see, particularly when the mice are awake and eating, they have increases in oxygen consumption. But particularly in the cold condition, you see that there's a really robust upregulation of these thermogenic responses. So what I've shown you thus far, just to summarize, to set the framework up for the next part of the talk, is that TMA and TMAO are diet-derived metabolites. We actually think of these as postprandial hormones almost. So in this case, and this is not really appreciated in the human literature because most of those are just fasted blood samples, but we see this both in humans and in mice. In this case, we fast mice, we gavage them with a single high-fat meal, and we see a spike of TMA, TMAO about 2 to 4 hours later. So this is likely substrate-driven, but there's a spike postprandially that goes down. And what we think is going on is when you eat a high-fat meal after a meal, this, and I'm going to introduce this microbial enzyme called CUT-C-CUT-D, which utilizes choline and converts it to TMA. We think that both TMA and TMAO are impacting these diseases through dedicated host receptor systems. So I don't have time to show you all the data, but there's a GPCR called trace amine associated receptor 5. It's only been studied in the olfactory bulb to this point, but it's what actually smells TMA. TMA smells like rotting fish, so it's a bacterial breakdown product that you would smell in the fish market. We also think once FMO3 converts TMA to TMAO that there's a host TMAO receptor. So the Hazen lab is trying to identify what that receptor is, and both of these signal in different cell types, macrophage, platelets, adipocytes, skeletal myocytes, to initiate these diseases. Okay, so the last part of the talk, I just want to highlight our efforts in drugging this pathway. So if this pathway is linked to all of these chronic diseases in people, why take pills that go to our human cells? Why not directly drug the enzymes in the bacteria with small molecules? So with this pathway in particular, there are a couple options of how to drug it. You could take antibiotics. That's never a long-term option because of antibiotic resistance and deleterious effects of chronic antibiotic exposure. You can reduce the dietary substrates, but you can never completely get rid of them. Phosphatidylcholine is in every diet, and it's also secreted in bile. So there's always substrate available to the bacteria. I showed you some of the data that you can inhibit the host enzyme FMO3. You do see some cardiometabolic disease benefit, but you don't necessarily want to inhibit this enzyme because this is a xenobiotic metabolizing enzyme, and it also converts TMA to TMAO. When TMA accumulates, in particular with human mutations in FMO3, you get something called fish odor syndrome, trimethylamine. So those people smell like rotting fish. So you don't necessarily want to inhibit this enzyme. That's not a desirable trait that you would want. We're doing very little with fecal microbial transplantation, but there's a lot of interest in this in infectious diseases like C. diff. Most of the pharmaceutical industry interest is in prebiotics or probiotics, so live culture bacteria or things in the diet which will help good bacteria grow. What I'm going to tell you about are our efforts here to selectively inhibit the microbial enzymes that take choline and convert it to TMA or carnitine to TMA. So Stan Hazen's group had this very recent paper, 2015 December, that showed basically they found a choline analog that binds to the active site of the microbial enzyme CUT-C, CUT-D, and it lowers the activity of the enzyme and lowers the level of TMA and TMAO. They fed it to ApoE knockout mice and they got less atherosclerosis. So it was a very simple paper, but it was one of the first proof of concepts that you could drug a bacterial enzyme with benefit to the host. And since then, Stan's group has a very talented group of synthetic chemists, which, building off the structure of DMB, have made a number of structural analogs. So this is just an inhibitory assay with the recombinant CUT-C, CUT-D. So here's the IC50 of DMB in the 10 micromolar range. Some of these lead compounds have picomolar IC50s. And they have very interesting properties in that they're largely covalent inhibitors. So they're binding to the active site in the bacteria, and they never let go. So they're actively incorporated and they're disposed of in the feces. So this is just a single gavage of one of the lead compounds, iodomethylcholine. This was just recently published a couple months ago. And you can see it's very effective in lowering TMA, TMAO up to 72 hours. So a single gavage, because it's a covalent inhibitor, you get massive reduction. But you get very little systemic distribution of the drug. So this plasma levels in the black, this is fecal levels of the drug. So the drug builds up in the bacteria and it's disposed of in the feces, lowers the production of the metabolite. The question is, what does it do? I'm going to skip this one. And in the original paper, Stan Hazen's group has shown that this pathway is very important in thrombotic responses. So this is an in vivo mouse assay where one of the arteries is injured with ferric chloride, and then the mice are injected with fluorescently labeled platelets. So this bright spot are platelets adhering to that site of injury. So when you feed choline, you get enhanced platelet accumulation and thrombus formation. And as a readout, the occlusion time, so the vessel is actually occluded within a short time frame. So when you feed choline, there's a shorter time to occlusion. The mice have a prothrombotic response. But if you pre-treat with either of these drugs, you completely prevent that effect. And then my lab has been more interested in the anti-obesity, anti-diabetic effects of these drugs. So in this case, what we did is we incorporated one of the lead compounds in a high-fat diet. So this just shows that plasma TMA, TMAO levels are down a lot with the drug. The mice eat the same amount throughout the study, but they weigh substantially less. They have improvements in glucose tolerance, and they also have modest reductions in hepatic triglycerides. This is another study where we incorporated one of these TMA lyase inhibitors into a chow diet, and these are OB mice. So these are hyperphagic mice that can't stop eating, so they become morbidly obese. So you can see the drug worked really well here. We have no detectable TMA, TMAO. Plasma choline levels are not affected. And actually the drug-treated mice eat more throughout the study, which is hard to do in an OB mouse, but they eat substantially more. But they weigh a lot less, in some cases 15 grams less at necropsy. So this is a very striking phenotype in OB mice. There are very few things other than leptin that can make a OB mouse lean. And we think this is all due to a massive increase in energy expenditure. So this is just oxygen consumption in metabolic cages. So I'll just bring it back together. So we think that when you and I eat a meal, there are substrates in that meal that are exclusively metabolized by gut microbes, providing this postprandial pulse in TMA, TMAO. We think after a meal that these host receptors are important in a tissue-specific way, cell type restricted way, and sensing those metabolites to drive a number of chronic diseases associated with overnutrition. And I will end with a few comments about this Center for Microbiome and Human Health. So really the focus is less on what types of microbes are there. I mean, you have to look at that just to have the descriptors in place, but we're much more interested in what are the gut microbes making. So what are the small molecule metabolites that are made only from gut microbes, and are they associated with disease? And I wanted to put this up because we can help you measure a number of these. We have within our microbial metabolomics core targeted panels to measure just about everything on this list, and several other classes, in particular a number of amino acid panels, which are quite expansive. So please email me if you have interest in collaborative studies. So with that, I will acknowledge lots of people that have helped. My two talented postdocs, Rebecca Sugar, Manya Warrior, have driven most of the FMO3 and TMA lyase inhibitor work. I moved to the Cleveland Clinic in 2013 to join forces with Stan Hazen and his group. It's been an amazing road, and we really think that this drugging of the TMAO pathway is one example of potentially many of those other metabolite pathways that come from gut microbes. But we have kind of a pipeline of moving through this and making drugs to target the bad actors. Jake Lucis, Peter Tontinos helped us with some of the early FMO3 stuff. Ionis Pharmaceuticals provided the ASOs. Michael Fischbach is collaborating with us on crosstalk between the TMAO pathway and bile acid metabolism. And then Jörg Kieren's group in Hamburg helped us with some of the adipose browning and beiging phenotypes. And of course, the funding for the research. So with that, I will stop and hopefully have time for questions.
Tenemos 5 minutos para preguntas. Si alguien tiene alguna pregunta, a favor de levantar su mano.
Hi, good morning. Nimbe Torres from the National Institute of Nutrition. I would like to ask you, have you seen association with the TMAO production, some specific bacteria, and LPS production?
So a number of groups have looked at what bacterial species, genus, order are associated with production. There are many, particularly with the choline, the cut C utilization cluster. It spans a number of taxa, so it's not like one bacteria is the producer. The way that these enzyme inhibitor screens were actually across a broad array of diverse phylogenetic species. And your second with the LPS?
Yes, LPS. Have you seen after the consumption of the high-fat diet, you see an increase in TMAO and TMAO? And have you seen an increase in LPS production?
So we've looked at this in a couple studies. We haven't looked in the drug study. I more so think that LPS is more of a general readout of leaky gut. So bacteria actually making it past the gut barrier, where TMAO is much more dietary derived. So there may be some coordinate, the bacteria that get in are engaging TLR4 at the same time, but I think of it as a different pathway, probably parallel pathway.
Okay, thank you.
Hi, very nice talk. I would like to ask you, it looks like TMA or TMAO are like inhibitors of browning.
Mm-hmm.
And do you know why?
This is something we've been working on for a couple of years. The original finding that we had is if we knock down FMO3, you get massive beiging of classic white adipose depots. So gonadal, perirenal, inguinal, subcutaneous fat pads turn brown-like. And they have this huge increased energy expenditure. We also have found that FMO3 is highly regulated by cold. So Jörg Kieren, for instance, put mice in cold 4 degrees for a week or 7 weeks, and there's dramatic, several thousand-fold upregulation in the liver, but reciprocal downregulation in adipose tissue. So I wish I could tell you we understand it. We do think it's a powerful regulator of host thermogenic responses, but I think it's coordinated both through liver and adipose tissue FMO3. I didn't show you the data, but we had a collaborator, Markku Laakso, in Finland, who has a huge population study where he has human adipose, subcutaneous adipose biopsies from about 1,000 men, and he looked at microarray levels of FMO3 and metabolic traits. So the more FMO3 you have in adipose tissue associates with BMI, obesity phenotypes, and negative responses with insulin sensitivity, but it also is negatively correlated to all of these brown beige marker genes, UCP1. So even in humans, we think that there's some kind of negative regulation of browning, beiging phenotypes. We're actively studying it, but I don't have a clear answer. We're looking at knockout mice of the receptor for TMA. We actually think TMA may be a signal that's a part of this, but we don't have the data yet.
Thank you.
It is possible to postulate the study of microbiota metabolomics in terms of identifying metabolites that predict incidence?
Yes. So
And if the panels will be, I mean, the metabolomic profiling will be different than
correct.
for the bacteria and the ones that we have.
So this is a big driving force of our discovery of these pathways, is to start with highly annotated clinical studies where you, especially when you have incident phenotypes. So we're doing this through both untargeted and targeted approach. So the untargeted analysis, like Stan Hazen's identification of TMAO, gives you power to look at everything unbiasedly, but the problem is it's hard to identify those unknowns, particularly if you have a mass charge ratio where there's 300 things that you're guessing on. We also have targeted panels for short-chain fatty acids, secondary bile acids, amino acid metabolites. They're all different platforms, like you mentioned, LCMS, GCMS, but those are the kind of things that we would be very interested in collaborating on to help you move your science forward. And that's the most advanced part of this core, is the metabolomics capability, because we think that's where you start. You understand the disease-associated metabolites, and then you have to go back to mouse studies to understand, are they causative, are they, and can you drug the pathway?
Nice studies try to produce differences in diet or
Yes.
are the way to handle this.
So that's the next step in the pipeline, is to basically feed the substrates in mouse diets with or without, ideally, the bacterial enzyme if you know it. And we actually have a group that are predictors of enzymatic capabilities. So they're more bioinformatics, reference genome mining folks that can predict that this particular gene would make a phenyl ring, or a particular chemical moiety within that metabolite. Mm-hmm.
We want to express our gratitude for your presentation. It was very clear and state of the art. Thank you so much.
Yeah, thank you for having me.